What's this? Ooh. It's a DNA structure. Cool. It looks like a X with bases. Speaking of DNAs, I'm going to tell you about DNAs and RNAs. DNA, abbreviation of deoxyribonucleic acid, is a molecule composed of two chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. Organic chemical of complex molecular structure that is found in all prokaryotic and eukaryotic cells and in many viruses. DNA codes genetic information for the transmission of inherited traits. 17 non-canonical bases are known to occur in DNA. Most of these are modifications of the canonical bases. They are N6-carbamoyl-methyladenine, N6-methiodinine, 7-methylguanine, N4-methylcytosine, 5-carboxylcytosine, 5-formylcytosine, 5-glycosylhydroxymethylcytosine, 5-hydroxycytosine, 5-methylcytosine, alpha-glutamitymidine, alpha-putrescinyltimine, base J, uracil, 5-dihydroxypentoracyl, 5-hydroxymethyldeoxyuracil, deoxyarcheosine, and 2,6-diaminopurine. DNA and RNA were first isolated by the Swiss physician Friedrich Miescher who, in 1869, discovered a microscopic substance in the pus of discarded surgical bandages. As it resided in the nuclei of cells, he called it nuclein. A nucleic acid sequence is a succession of letters that indicate the order of nucleotides forming alleles within a DNA, using GACT, or RNA, GACU, molecule. Nucleic acid secondary structure is the base pairing interactions within a single nucleic acid polymer or between two polymers. It can be represented as a list of bases which are paired in a nucleic acid molecule. Nucleic acid tertiary structure is the three-dimensional shape of a nucleic acid polymer. RNA and DNA molecules are capable of diverse functions ranging from molecular recognition to catalysis. Nucleic acid quaternary structure refers to the interactions between separate nucleic acid molecules, or between nucleic acid molecules and proteins. The concept is analogous to protein quaternary structure, but as the analogy is not perfect, the term is used to refer to a number of different concepts in nucleic acids and is less commonly encountered. The double helix is the dominant tertiary structure for biological DNA, and is also a possible structure for RNA. Just like in biology, biological DNA, or DNA, is a biological macromolecule that carries hereditary information in many organisms. DNA exists in many possible conformations that include aDNA, bDNA, cDNA, ddNA, eDNA, LDNA, pDNA, sDNA, and zDNA. They are all forms of DNA in terms of structure and environment. Environmental DNA, or EDNA, is a DNA that is collected from a variety of environmental samples such as soil, seawater, snow or even air rather than directly sampled from an individual organism. As various organisms interact with the environment, DNA is expelled and accumulates in their surroundings. It is present in every cell at a very high relative humidity, 92%, and low concentration of ions. It has anti-parallel double helix, rotating clockwise, right hand, and made up of sugar phosphate backbone combined with base pairs or purine pyrimidine. The A form of DNA is found at 75% relative humidity in the presence of sodium, potassium, or cesium ions. It contains 11 base pairs as compared to 10 base pairs of B DNA which tilt from the axis of helix by 20.2 degrees. The C form of DNA is found at 66% relative humidity in the presence of lithium and magnesium ions. As compared to A and B DNA, in cDNA the number of base pairs per turn is less, that is 28/3 or 9 1/3. The base pairs show pronounced negative tilt by 7.8 degrees. The D form of DNA is found rarely as extreme valence. Total number of base pairs per turn of helix is 8. Therefore, it shows 8-fold symmetry. This form is also called poly, DDT, and poly, DGDC, form. There is pronounced negative tilt of base pairs by 16.7 degrees as compared to C form. The eDNA is also known as eccentric DNA. eDNA has a long helical axis rise and base perpendicular to the helical axis. It has a deep major groove and shallow minor groove. eDNA allowed to crystallize for a period time longer, the methylated sequence forms standard aDNA. eDNA is the intermediate in the transition to aDNA. The zDNA has been found in a large number of living organisms including mammals, protozoans and several plant species. The double helix makes one complete turn about its axis every 10.4 to 10.5 base pairs in solution. This frequency of twist, termed the helical pitch, depends largely on stacking forces that each base exerts on its neighbors in the chain. The absolute configuration of the bases determines the direction of the helical curve for a given conformation. It is a right-handed double helix fairly similar to the more common bDNA form, but with a shorter, more compact helical structure whose base pairs are not perpendicular to the helix axis as in bDNA. It was discovered by Rosalind Franklin, who also named the A and B forms. She showed that DNA is driven into the A form when under dehydrating conditions. Such conditions are commonly used to form crystals, and many DNA crystal structures are in the A form. DNA and RNA are nucleic acids, alongside proteins, lipids and complex carbohydrates, polysaccharides. Nucleic acids are one of the 4 major types of macromolecules that are essential for all known forms of life. RNA, also known as ribonucleic acid, is a polymeric molecule essential in various biological roles in coding, decoding, regulation and expression of genes. RNA and DNA are nucleic acids, and, along with lipids, proteins and carbohydrates, constitute the 4 major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, it is more often found in nature as a single strand folded onto itself, rather than a paired double strand. Cellular organisms use messenger RNA to convey genetic information, using the nitrogenous bases of guanine, uracil, adenine, and cytosine. Messenger RNA, or mRNA, is a large family of RNA molecules that convey genetic information from DNA to the ribosome, where they specify the amino acid sequence of the protein products of gene expression. RNA polymerase transcribes primary transcript mRNA into processed, mature mRNA. Pre-mRNA is synthesized from a DNA template in the cell nucleus by transcription. Pre-mRNA comprises the bulk of heterogeneous nuclear RNA. Heterogeneous nuclear RNA, or HNA, is a mixture of RNA, of high molecular weight, that never leaves the nucleus, has a high rate of turnover and consists of mRNA containing both introns and exons of genes. A primary transcript is the single-stranded ribonucleic acid, RNA, product synthesized by transcription of DNA, and processed to yield various mature RNA products such as mRNAs, tRNAs, and RNAs. A transfer RNA, abbreviated DNA and formerly referred to as SNA, for soluble RNA, is an adapter molecule composed of RNA, typically 76 to 90 nucleotides in length, that serves as the physical link between the mRNA and the amino acid sequence of proteins. DNA does this by carrying an amino acid to the protein synthetic machinery of a cell as directed by a 3-nucleotide sequence in a messenger RNA, mRNA. Soluble RNA is another name for transfer RNA. Ribosomal ribonucleic acid, RNA, is the RNA component of the ribosome, which is essential for protein synthesis in all living organisms. Mammalian cells have 2 mitochondrial, 12S and 16S, RNA molecules and 4 types of cytoplasmic RNA. Complementary RNA, or CRNA, is a viral RNA that is transcribed from negative sense RNA and serves as a template for protein synthesis. Negative sense RNA is a viral RNA with a base sequence complementary to that of mRNA. During replication it serves as a template for the transcription of viral complementary RNA. Positive sense RNA is a viral RNA with the same base sequence as mRNA. During replication it functions as mRNA, serving as a template for protein synthesis. Small nuclear RNA, or SNA, is a class of eukaryotic small RNA molecules found in the nucleus, usually as ribonucleoproteins, and apparently involved in processing heterogeneous nuclear RNA. Antisense RNA, or SNA, is a single-stranded RNA that is complementary to a protein-coding messenger RNA with which it hybridizes, and thereby blocks its translation into protein. As RNAs, which occur naturally, have been found in both prokaryotes and eukaryotes. Antisense transcripts can be classified into short, 200 nucleotides, and long, 200 nucleotides, non-coding RNAs. A non-coding RNA, or CNA, is an RNA molecule that is not translated into a protein. The DNA sequence from which a functional non-coding RNA is transcribed is often called an RNA gene. Abundant and functionally important types of non-coding RNAs include transfer RNAs and ribosomal RNAs, as well as small RNAs such as microRNAs, siRNAs, pRNAs, snRNAs, SNAs, xNAs, scarNAs and longNAs such as XIST and HOTER. A microRNA, or mRNA, is a small non-coding RNA molecule, containing about 22 nucleotides, found in plants, animals and some viruses, that functions in RNA silencing and post-transcriptional regulation of gene expression. Small interfering RNA, or siRNA, is a class of double-stranded RNA molecules, 20-25 base pairs in length, similar to mRNA, and operating within the RNA interference pathway. It interferes with the expression of specific genes with complementary nucleotide sequences by degrading mRNA after transcription, preventing translation. Pwi-interacting RNA, or pRNA, is the largest class of small non-coding RNA molecules expressed in animal cells. pRNAs form RNA protein complexes through interactions with Pwi proteins. These pRNA complexes are mostly involved in the epigenetic and post-transcriptional silencing of transposons, but can also be involved in the regulation of other genetic elements in germline cells. pRNAs are mostly created from loci that function as transposon traps and provide an RNA-mediated adaptive immunity against transposon expansions and invasions. They are distinct from microRNA, mRNA, in size, 26 to 31 nt rather than 21 to 24 nt, lack of sequence conservation, and increased complexity. Small nucleolar RNAs, or SNRNAs, are a class of small RNA molecules that primarily guide chemical modifications of other RNAs, mainly ribosomal RNAs, transfer RNAs and small nuclear RNAs. There are two main classes of SNRNA, the CD-box SNRNAs, which are associated with methylation, and the HACA-box SNRNAs, which are associated with pseudoradylation. SNRNAs are commonly referred to as guide RNAs but should not be confused with the guide RNAs that direct RNA editing in trypanosomes. Extracellular RNA, or exNA, is also called exosomal RNA. It describes RNA species present outside of the cells from which they were transcribed. In Homo sapiens, exNAs have been discovered in bodily fluids such as venous blood, saliva, breast milk, urine, semen, menstrual blood, and vaginal fluid. Although their biological function is not fully understood, exNAs have been proposed to play a role in a variety of biological processes including syntrophy, intercellular communication, and cell regulation. Small Kugel body-specific RNAs, or sRNAs, are a class of small nucleolar RNAs that specifically localize to the Kugel body, a nuclear organelle involved in the biogenesis of small nuclear ribonucleoproteins. RNA is the predominant RNA in most cells, composing around 80% of cellular RNA. Ribosomes are approximately 60% RNA and 40% protein by weight, or 3/5 of ribosome mass. Prokaryotic ribosomes contain 2 major RNAs and 50 or more proteins. Ribosomes form 2 subunits, the large subunit and small subunit. The LSU acts as a ribozyme, catalyzing peptide bond formation. The SSU decodes the mRNA. SSU and LSU sequences are widely used for working out evolutionary relationships among organisms, since they are of ancient origin and are found in all known forms of life. Mature messenger RNA, often abbreviated as mature mRNA, is a eukaryotic RNA transcript that has been spliced and processed and is ready for translation in the course of protein synthesis. A precursor mRNA, or pre-RNA, is a type of primary transcript that becomes a messenger RNA, mRNA, after processing. Pre-mRNA is synthesized from a DNA template in the cell nucleus by transcription. Pre-mRNA comprises the bulk of heterogeneous nuclear RNA. RNA interference, or RNA, is a biological process in which RNA molecules inhibit gene expression or translation, by neutralizing targeted mRNA molecules. Historically, RNA was known by other names, including co-suppression, post-transcriptional gene silencing, and quelling. The detailed study of each of these seemingly different processes elucidated that the identity of these phenomena were all actually RNA. Mature mRNA is also called mature transcript, mature RNA or mRNA. The 2 DNA strands are also known as polynucleotides as they are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of 4 nitrogen-containing nucleobases, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. The nitrogenous bases of the 2 separate polynucleotide strands are bound together, according to base pairing rules, with hydrogen bonds to make double-stranded DNA. The complementary nitrogenous bases are divided into 2 groups, pyrimidines and purines. In DNA, the pyrimidines are thymine and cytosine. The purines are adenine and guanine. Adenine is a nucleobase along with guanine, cytosine, and thymine in the DNA, and adenine, uracil, guanine, and cytosine are in the RNA. Both strands of double-stranded DNA store the same biological information. This information is replicated as and when the 2 strands separate. A large part of DNA, more than 98% for humans, is non-coding, meaning that these sections do not serve as patterns for protein sequences. The two strands of DNA run in opposite directions to each other and are thus anti-parallel. Attached to each sugar is one of 4 types of nucleobases, informally, bases. It is the sequence of these 4 nucleobases along the backbone that encodes genetic information. RNA strands are created using DNA strands as a template in a process called transcription. Under the genetic code, these RNA strands specify the sequence of amino acids within proteins in a process called translation. Within eukaryotic cells, DNA is organized into long structures called chromosomes. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, providing a complete set of chromosomes for each daughter cell. Eukaryotic organisms, like animals, plants, fungi and protists, store most of their DNA inside the cell nucleus as nuclear DNA, and some in the mitochondria as mitochondrial DNA, or in chloroplasts as chloroplast DNA. Mitochondrial DNA, or mRNA, is the DNA located in mitochondria, cellular organelles within eukaryotic cells that convert chemical energy from food into a form that cells can use. Nuclear DNA, or RNA, is the DNA contained within each cell nucleus of a eukaryotic organism. Nuclear DNA encodes for the majority of the genome in eukaryotes, with mitochondrial DNA and plastid DNA coding for the rest. Nuclear DNA adheres to Mendelian inheritance, with information coming from 2 parents, one male and one female, rather than matrilineally, through the mother, as in mitochondrial DNA. Chloroplast DNA, or CPNA, is a DNA located in the chloroplast. It is also known as the plastome when referring to genomes of other plastids. Its existence was first proven in 1962, and first sequenced in 1986, when 2 Japanese research teams sequenced the chloroplast DNA of liverwort and tobacco. Since then, hundreds of chloroplast DNAs from various species have been sequenced, but they are mostly those of land plants and green algae. Glaucophytes, red algae, and other algae groups are extremely underrepresented, potentially introducing some bias in views of typical chloroplast DNA structure and content. In contrast, prokaryotes, bacteria and archaea, store their DNA only in the cytoplasm, in circular chromosomes. Within eukaryotic chromosomes, chromatin proteins, such as histones, compact and organize DNA. These compacting structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. DNA was first isolated by Friedrich Miescher in 1869. Its molecular structure was first identified by Francis Crick and James Watson at the Cavendish Laboratory within the University of Cambridge in 1953, whose model building efforts were guided by X-ray diffraction data acquired by Raymond Gosling, who was a postgraduate student of Rosalind Franklin. DNA is used by researchers as a molecular tool to explore physical laws and theories, such as the ergodic theorem and the theory of elasticity. The unique material properties of DNA have made it an attractive molecule for material scientists and engineers interested in micro and nanofabrication. Among notable advances in this field are DNA origami and DNA-based hybrid materials. There's also prokaryotic and eukaryotic DNA. The key difference between prokaryotic and eukaryotic DNA is that the prokaryotic DNA freely floats in the cytoplasm while the eukaryotic DNA situates inside the nucleus. The reason for that because prokaryotes have a simple cellular organization. They lack a nucleus and membrane-bound organelles. On the other hand, eukaryotes have a complex cellular organization. They have a true nucleus that contains DNA and also membrane-bound cell organelles. All the prokaryotes are unicellular while eukaryotes may be unicellular or multicellular organisms. Prokaryotes and eukaryotes mainly contain DNA genomes. Their DNA is packaged into chromosomes. Since prokaryotes do not have a nucleus, their DNA freely floats in the cytoplasm. However, in eukaryotes, chromosomes situate inside the nucleus. Hence, nuclear membrane encloses all eukaryotic DNA. DNA and RNA both collect information. That's all I want to talk about DNA and RNA. This is Iggy Koopa, signing off. Oh, and one more thing. On May 6, 2019, Vyond announced the removal of the legacy video maker, due to Flash ending support. The legacy video maker removal was going to be on January 1, 2020. Just because the legacy video maker shut down, does not mean that the whole website shuts down. After that date, you need to move on to the new video maker, which is compatible, and supports HTML5. But that means you won't be able to make grounded videos anymore. So, we will going to miss the legacy video maker when January 1, 2020 comes. Now this is Iggy Koopa, signing off.